Off only.
Yes,
Or you want,
sir. Anything?
yeah. So maybe during question answer you can keep the video on. That will be better.
Okay, sir.
So good afternoon, everyone. So I'm happy to share some of our recent experiences on signal processing. Actually, we have been working on the detection of cardiovascular diseases, and we have not really been looking at it as a signal processing activity. But when the request came from Signal Processing Society, thinking, and I felt that yes, most of what we are doing is basically signal processing, and of course, they could sometimes be called high-level signal processing, including some classification and deep learning as well coming into this picture. So let me try to present our work that we have been doing in the recent past uh, to all of you. So this is a brief organization. So first I'll talk about what are our cardiovascular systems and what are the major diseases and why they are a kind of threat for us. And then we'll talk about some of the ways in which we can estimate the blood pressure. And blood pressure is a very, very important measurement to be made. And anybody having hypertension or other diseases, immediately doctors immediately take their blood pressure. Basically, what the doctors do is basically they take two measurements. They call it as systolic blood pressure and diastolic blood pressure, right? So we'll be using these terms again and again. SBP and DBP, standing for systolic and diastolic blood pressures. Like normal people will be having 110, 70, or 120, 80. These are typically in the normal range. When we say that you don't have a hypertension, right? So I'll talk about how that could be that have been estimated over the years and what are the current approaches. And these days we actually try to estimate not only these two quantities which are called systolic and diastolic blood pressure, but what we actually call as a blood pressure waveform, which looks like this. So typically a blood pressure waveform it looks like this, where the highest topmost place is called the systolic blood pressure and the bottom part is called the bottommost part is called the diastolic blood pressure. So typically using the sphygmomanometer technique which doctors use, they can actually measure these two levels by listening to the oscillations within the manometric system. And people have been satisfied for many years with these two measurements because nothing better could be done. But more recently, there is a and there is an interest to actually estimate this blood pressure signal, right? So when we say signal, is basically this type of a signal, and that is actually not such an easy task, right? Especially as I explained to you, that there are some more restrictions put in, like we don't want it to be an invasive measurement. In fact, the so-called arterial blood pressure can be measured only by some invasive process where you actually will have some probes put into your arteries, and from there only you can actually measure the blood pressure, just like we can measure the pressure of a water supply or rate of flow of water by just putting a manometer uh, in the pipe. Uh, so that kind of instrument actually we cannot put actually in human bodies just like that. So we'll talk about that, and then we'll describe some of the approaches that we have tried, and some of the approaches, that is basically we have also tried to assess the existing approaches, and we did not really feel them to be very powerful as of now. But we'll talk about it, and then we'll share what are the things that we have been trying to do uh, in our case. And then I'll go a little bit deeper, and there will be reason, as we'll explain these two problems, I'll explain to you why we need to go a little bit deeper into the cardiovascular system, that we have to understand what is happening inside the body as we make some external measurements. Modeling is very, very important. And when we model it, then we can actually use that to detect various diseases like the coronary artery disease. And we will call it again with an abbreviation CAD, CAD. And then we'll talk about some more signal processing and how we can detect cardiac abnormalities using the so-called electrocardiograph signal ECG or phonocardiogram signals, the PCG, right? So I'll try to cover the gamut of all these signals and uh, finally try to conclude with recent trends and what I'll call as fashions, and then try to and give you some references as well. So to give you history, so why cardiovascular diseases are important? As you can see, since 2000 and probably even before that, and even more recently, you can see that cardiovascular disease is actually the number one reason for death. In fact, the number of people who die of cardiovascular disease, in fact, has increased uh, over these last 20 years or so. And in fact, of course, these reasons like cancer also, it has also increased. Overall, it has increased, but in fact, it has increased more. On the other hand, respiratory diseases have been managed in a little bit better way, so there is a decrease. But neurological disorders again have gone up, right? And kidney diseases and all that, and diabetes and all, they have also gone up. So many of them have gone up, but cardiovascular disease still hold number one. So this is the worldwide figure, and Indian figure is not different. In fact, you will see that the Indian, in fact, the increase in cardiovascular disease, the increase is even more, right? It's almost 30% increase over the last 2 decades, right? And here, respiratory diseases have been more. In fact, it was number two here, still number two, but it is percentage wise little bit it has gone down. On the other hand, cancers, diabetes, etc., they are all rising, right? So these are all rising. So it is very important to control the cardiovascular diseases, and doesn't have a strong cardiovascular system, many other diseases can happen. So that's also an issue. So it is like, the blood is flowing through the body and it is carrying oxygen. So if it is not happening properly, a lot of other portions in the body will starve. In fact, in the era of COVID-19, as you all know, that people are those weaker cardiovascular systems are said to be more susceptible and are apparently having more difficulties to recover. So it's very important to have a strong heart, right? And CAD is again a particular cardiovascular disease, right? And so that also is a serious threat. And since I'm going to talk more about CAD, especially in terms of disease detection. So let me mention that both worldwide as well as countrywide, you can see that 16% of the people, both worldwide and countrywide, uh, they were vulnerable, susceptible to CAD. And stroke is also another disease due to a cardiovascular problem also, in the sense that blood flow in the brains, actually, or lack of blood flow in the brain can cause a stroke. So these two together constitute almost 27% of the all the life-threatening diseases. So how do we go about it? So there are a lot of signals that we can actually use for detecting diseases. Especially we are interested to detect diseases before they become life-threatening, right? So early detection is always very, very welcome. So if we have our, if we consider the human heart and the cardiovascular system at the center, a few signals which have been always been used for detection of diseases is the let's say the first one that comes to our mind. In fact, the doctor will immediately order an ECG to you if you are going to have high blood pressure and you have some chest pain or something. It's not that it can actually detect diagnose all the diseases, but it can actually give you an indication. So there are many diseases. I'm not going into the details of all the names, right? In fact, I may not be even able to pronounce all of them without any problem, because the literature. right? So there are various types of malfunction of the heart, right? And since we work on coronary artery disease, we are a little bit more familiar with it. So basically, artery disease is if you have some kind of plaque formation in your arteries, right? And especially the coronary arteries, which is providing blood to the heart, right, for the pumping the heart muscles, right? So that kind of blockage in those coronary arteries, in fact, anybody having some angioplasty, etc., would probably be having it in the coronary artery only, because that is where it becomes a problem, because heart has to pump blood to the entire body, but heart has to be operated by the coronary, I mean, that has to be supplied by the coronary artery. So if there is any disease, it is like the, like the muscles of the heart itself do not work, so that creates a big issue. So I mean, a lot of efforts have been made. In fact, even physical and mental stress can be detected from ECG. The another one, which is conventionally used by doctors. In fact, doctors listen to the heart, right? So they put their stethoscope in the ears and then they listen to what is happening. In fact, they can actually detect lots of diseases, especially what are called, so any valve is not working. By listening to the sound of the heart, they can figure out whether there is a valvular or valve disease, right? Whether there are murmurs, right? We'll come to these kind of things even more. In fact, we'll have a section on PCG where we'll talk about how many of them are, those are getting automated, right? And also coronary artery disease, and this is increasing. In fact, these days the tendency is always try to do things with these kind of signals, which are so-called non-invasive, right? You just listen, right? You don't disturb anything in your heart. You just put a sensor close to your heart. Sometimes you don't need to even put it on the body. You can just keep it close to it, right? And if you can get a signal from there, there's nothing like it, because even pre-COVID days we wanted non-invasive treatment, and post-COVID days I'm sure nobody would like them to be touched unnecessarily. And then there is a signal, which is the one I talked about in the beginning, the so-called arterial blood pressure signal. So this arterial blood pressure signal is invasive, right? That is, you have to put some probes inside your body to measure it. But the good thing about it is that you can actually detect various things like blockage or stenosis, right, or various irregularity in the heartbeats and things like that, right? And atherosclerosis, which is again a kind of a plaque formation all over the arteries in the system, and again many other things, uh, as the doctors describe them in this fashion, right? So lots of diseases are actually possible to be identified if you can actually get the blood ABP signal. And that is the reason we'll try to figure out how to get that ABP signal from other kind of non-invasive measurement, right? So if you can actually get this ABP signal by kind of combining signals from ECG, PPG, and maybe other signals, right? Then we can actually access all these, detect all these diseases. So that is the thing that we are all trying to do. So, so if we just have the PPG, so-called photoplethysmogram signal, in fact, if you go to the emergency of a hospital, you will see that they'll just put a probe on your fingers. I'll give you a picture of that also. On the fingers, they will put a probe, and that will actually give you the blood pressure, at least systolic, diastolic blood pressures and things like that. And from here also, this signal is now actually is very much in the sort of news, because you can actually, in fact, as I will show, that you can actually get ABP signal, kind of predict the ABP signal if you can have it, maybe along with some other ECG signals and things like that. And once you can do that, once you can estimate this ABP signal, whatever ABP can do, PPG can also do. Right? And PPG is non-invasive. Non-invasive. In fact, all these ECG, ECG, and PPG. These are all non-invasive to some extent, and then I'll explain in another chart that in fact these days the tendency is to have signals which are non. In fact, these are inobtrusive, right? You do not even know that they are actually there, and they'll still can get the signals. So these are the gamut of cardiovascular signal processing so far that I've come across, and so we can do a lot of things once we get the these measurements properly. So as I was telling that there have been a lot of instruments, especially earlier days instruments used to be invasive instruments, right? You put some probes inside the body and then you measure the blood pressure, right? And then there came this so-called arterial line. So in your wrist, right, you have arteries. So if you can put a probe by making a little incision to there, if you can measure the blood pressure, then you will get an exact blood pressure at that point. Right? But then again, this could be done only in an ICU type of environment when you are admitted to the hospital. Now, you would not like your blood pressure to be taken in this fashion if you are just going for a casual visit. Right? So from 60s, we have these so-called cuff-based measurements, right? Wrist cuffs, right? Arm cuffs, or even finger cuffs are there. So basically, what is done is the same principle which doctors use for measuring the blood pressure is automated, right? By a method called tonometry. Basically, doctors listen to the tone of the signal. So this can be also automated by using a method where you can have you can apply pressure from a controlled source and then slowly raise the blood pressure, external pressure, and then whenever it matches the systolic and the diastolic blood pressure, then the tone changes. So the instrument can detect that. And then in the 70s, a method called volume clamp. In fact, we'll go into a little bit more detail about it, because this is there is an instrument called Phenopress which uses this technique. There is a patented technique where you can put a finger, it's a pulse plethysmogram signal, sensor in your finger, and then by adjusting the pressure on your finger, you can measure the pulse plethysmogram signal in a quite accurate fashion, and from there you can derive many other quantities like the arterial blood pressure, cardiac output, and various other things, right? And of course, these days we are trying to have more cuffless, right? So we'll see whether that is possible or not. And still, I do not think any technique is there where it's really fully cuffless methods are working. In fact, this Phenopress method comes close to it, where there is a finger cuff, but time to time it needs to do a calibration so that it does not go out of calibration with the blood pressure, and so that creates a gaps in the measurement, especially when you are interested to find the arterial blood pressure signal, right? And then we have to figure out ways to fill up those gaps. So I'll talk about that in an example. And this is a little bit of an overview of some of the recent approaches. So if you have multiple signals like PPG, ECG, and sometimes you also add your knowledge about the system in the form of a model, right? And people try to estimate the arterial blood pressure from these signals. Sometimes people take two places. They take PPG signal, plethysmogram signal, from two places in the body, and then they get quite a good accuracy, especially if they combine that with the ECG signal. Then one of the recent work, basically they combined the effect of basically they get so-called PAT signal. I'll show you a diagram where these are all explained in a better way, and heart rate, right? And so they could measure it quite accurately. Then there are other approaches where you use a model and a PPG signal, or you use some other model, Two different papers have been there in the recent past. In fact, this one uses some deep learning CNN-based method. And then we have another one where actually you take the PPG signal and construct what is called the pulse wave velocity. And there you then try to estimate the blood pressure. Right? And the accuracy of these, they vary. But the first one, especially, you will see that is a very has a very good accuracy, because if you have 100, the nominal values of your blood pressure, if they are in the order of 100, so if 1 or 2 millimeter variation means 2% variation, which is very, very good, right? But you can see that here actually you need 2 PPG signals and 1 ECG signal, so that is a lot of overhead. On the other hand, many others which use a single PPG signal, the accuracy is not that good, in the sense that they're more than 5%, right? And more than 5% is actually not acceptable from clinical terms. So if you build an instrument which is having this much of error, it will not be clinically acceptable. This one actually, of course, this is a deep learning based system. So according to the way that deep learning people, they write the accuracy, this happens to be 97%. But this is more like a classification problem that they have solved. So it is, written an error of 3%, but it is not a consistent error, right? What will happen is, in some cases the error may be large, in some other cases error will be small. So it is not the maximum error. Similarly, this one is again one PPG based measurement, but again you see that the RMS errors are quite high, right? Again, these two are of the same order, right? And which is what we have also experienced in our approaches, that the most of the approaches, their RMS errors in the so-called systolic blood pressure will be of the order of 8, 9 of that order. So according to American Association for Cardiologists, they have a standard of what is called, they call it the gold standard, which is that the error should be within plus minus 5 millimeters. So as I was telling, so what is this PAT or these kind of signals? So basically, this is how the various signals look like. So many of you would have come across this ECG signal, right? There is a peak, which is many of us actually refer to it as R peak, because what we do is actually we mark different points as Q, R, S and things like that, right? So P, Q, R, S, T, and like that we define the various parts of the ECG signal. So this R peak is a very important parameter, and it is very easily detectable, right? Because it's a huge peak. Depends on which lead you are looking at, but most leads will allow you to see this signal. And ECG, or the so-called phonocardiogram signal, looks like this. It is a little bit more noisy, but whatever you want to hear, right, that will happen in some parts which are called, so-called S1 peak and S2 peaks and things like that. Yeah, I hope I am getting it. So this is called S1 peak, and there is another peak which is called S2 peak, right? So these two places are very important to observe, right? And there are this is probably a healthy signal, so you don't see much oscillations. But especially with diseased people, there will be a lot of oscillations near the S1 peak and S2 peak. And by listening to that sound, doctors can figure out whether there is a disease. Now, this PPG signal or photoplethysmogram signal, right? So this is this signal actually looks similar to the arterial blood pressure, right? You will see that arterial blood pressure signal also, as I have drawn it, looks like this. In fact, PPG in a more pure form will also look like this, right? So there will be a peak, and there will be a trough, and in between there will be a notch type of thing, which is called dicrotic notch. Right. So in fact, many PPG signals, nice ones, will taken. They will also have a notch, right? So basically, the PPG to ABP, there is a lot of similarity, but we need to have, there will be factors and then there will be delays. Especially, you can see that the peak of the PPG comes much after the so-called R peak, right? This is the R peak, first R1, then you can say this is R2 and so on. So first R peak to PPG, so this gap is called PAT, right? It's the pulse arrival time. This is full form is pulse arrival time. And there is a small period which is the between the R1 peak and the S1 peak of the ECG is called the pre-ejection period, right? And this gap between the S1 peak to the peak of the PPG is called pulse transit time. Okay, I think it's not necessary to remember all this right now. And I'll come to specific cases where I'll define it as well. So this is just to give you a preview of the various signals that we come across in processing to be processed for detecting various diseases. So as I was stating that there is a relation between this blood pressure and this pulse transit time. Okay, so their formula actually there is an empirical relation where you can see that there is a pulse transit time and the blood pressure, right? So there is a logarithmic relationship between them with some coefficients, along with certain other parameters which depend on the of the person, right? So it looks like this. So pressure is equal to K1 times log of ETT plus some K2, where K2 is this function, right, which is a function of the length of the artery and various constants, density of the blood, elasticity, and things like that. So on an average, they more or less, they become a kind of offset, right? So what will happen is that from suppose, so you can see that if you are taking the measurement in some place in the US, right, there the size of the artery and the length of the artery, those things will be different in a normal person, right? And consider that with India, right? So what will happen is that these constants will be different potentially. And if you want to use this formula, you have to take care of it by doing sufficient experimentation in each of those countries. So you have to do properly, proper anthropological surveys and figure out what are the places where their body structures are different, and for each structure, what will be the kind of arterial sections that we are going to have. So what we tried to do is that we wanted to try it out from signals, right? And in fact, what we did is that we use the ECG and the PPG signals from data available from Physionet, right? Physionet. And then we tried to see whether this kind of formula is correct or not, right? How accurate it is. So we actually, from the signals PPG, we measured from ECG we got the heart rate, and from ECG and PPG we got the so-called PAT, right? Remember, we should have been using PTT, right, which is the actual formula. But since we did not have the PCG measurement, right, so we used the PAT, right? So since we have the ECG and the PPG, so we could only get the PAT, right? PTT, basically this pre-ejection period is unknown to us, right? So we had to use some alternative, whatever is available. Later on, we have also tried with PTT in place, but even that doesn't change things much. So once we have this, we try to find the correlation between these variables like heart rate, PAT, and blood pressure. And then we define some correlation coefficients, right? But unfortunately, we actually found that the correlation values are quite inconsistent, right? In fact, we had different subjects, right? 7 subjects here, and you can see PAT correlation versus SBP, DBP. They are quite erratic, right? And so is that with the heart rate as well, right? And there are different reasons for that. Firstly, for getting a good correlation, you need to measure these time periods accurately. But what we found later on that this PhysioNet database does not have accurate timing information. So even if ECG signal and PPG signals are available for the same patient, right, they are not time synchronized. So there is a scope of error. And then there is another issue, which is that if you have people in the ICU, right, because this PhysioNet data, PhysioNet contains a lot of data where patients are in ICU, right? So ICU patients are under medication, and their heart rates and their blood pressures, none of them are in the normal range. So it did not really give us a very satisfactory result. In fact, if you want to predict the systolic blood pressure and diastolic blood pressure, they come quite accurately. But the problem is that the error is not consistent and has a very funny kind of pattern, right? So this is so-called, Bland or Altman plot, where we actually plot the average value of the estimate versus the difference between the estimates. And ideally, it should be a random kind of plot, right? But you can see that this is having a pattern, kind of, linear pattern, both SBP and DBP. That means this is not a good estimation. So as I mentioned, so it's not very easy, although theoretically it's possible to calculate it from this formula, right? So if you have to measure the PTT, then you require 3 sensors. You need ECG, then you need PCG and PPG. And we also tried fitting the blood pressure as a nonlinear function of PAT, even that didn't help. So as I mentioned, so lack of synchronized data collection is actually the main reason for the poor correlation, right? And so what we felt is that we need to improve on this technique. And so one of the things that we tried more recently, so basically this was kind of a negative result that we published in last year's EMBC. And this year we tried to change our approach, and we tried to use a linear transfer functions approach rather than using a empirical formula type of approach. Right. So what we thought is that if we have our system, right, our physiological or cardiovascular system, right, and if you have both measured ABP and measured PPG, then can we not fit a transfer function model between them, right, with the measured ABP as the input and the PPG as the output, and then we'll minimize the error, and then we'll be able to estimate the transfer function, right? So basically, we are trying to model the process of estimating or measuring the PPG, right? So this can be also looked at like what is called as a Wind-Kessel model or compartmental model, where arterial blood pressure is actually kind of passing through a filter, and the filtered output is our PPG signal, right? So where these parameters, L, R, C, and all that will depend on the various factors like inertia of the blood, resistance of the blood flow, compliance of the artery, and viscoelasticity and things like that. So if we can actually know this kind of equivalent circuit, right, that is lying between the ABP and the PPG, then we should be able to reverse the problem, right, and then get solve the inverse problem, right? So if you know the model, right, so if your model is the ABP and you can get the PPG, so by inverting the model you should be able to get the ABP when PPG is given. So we tried this out, And these worked actually quite well. And this year actually Ashutosh has got another paper in EMBC. So where we actually estimated this transfer function. In fact, we tried to estimate it both ways, ABP to PPG and PPG to ABP. And as expected, these two transfer functions are actually inverses of each other. Right? The gain constants are inverses. This is this constant. There is a typo also here. So anyway, these all others you can see they are matching. Right? There is probably a zero missing somewhere from this. Okay, so you can see that if you have both ways, we have the models, then we should be able to predict one from the other quite accurately, and that's what happened, right? So this shows ABP as the input and PPG as output, right? And they are coming very close to each other, and this shows PPG as input and ABP as the output. They are also coming quite accurately. And so we did it for multiple subjects, right? And we determine the accuracy of training, accuracy of fitting and testing, right? And so two ways. One is the arterial modeling, which is ABP to PPG. And ABP estimation is from PPG to ABP. So you can see that these accuracies are not too bad. They're of the order of at least around 80 to 90%, right? Which is much better than the kind of correlation that we got earlier, right? And the sigmas are also reasonably small, right? So in fact we saw we have put it in the Bland-Ortmann form, right? And you will see that these are all flat, right? So this has a much better response, and you can see that the correlations are also quite good. So it's a linear fit, right? And that way we could get a much better response. And in the same way, for another subject, we could also see the same type of plot. So this is a recent paper that got published in EMBC this year. Now I'll come to something very kind of interesting that we came across while using the data from a FinaPress instrument. So as I've been mentioning, this is an instrument which actually helps us to estimate ABP signals from PPG signal. First, I'll explain what is the approach that is used and what are the limitations. So basically, what is done is that we put a IR probe on the finger, right? Infrared LED is put, and then there is a detector is put, right? And so we can actually see the kind of the optical output actually depends on the arteries which are there in the finger, right? So with blood pressure, the size of the artery keeps increasing and decreasing, right? So this pulse plethysmogram signal or the infrared optical signal, the output changes with the size of the artery, right? As arteries flatten, they get expanded, you get a variation, expanded and contracted, you get a variation in the optical output, right? So if it is uncontrolled, right, then the variations can be shown in this fashion, right? PPG signal. But what happens, and how actually it is used to estimate the blood pressure is that by putting an external force or pressure, by an external pressure along with the IR sensor, if you could actually manage to track this variation, right, then we can actually minimize these oscillations, right? So if we can keep it, if we can actually stop the oscillations by keeping the arteries, by balancing the internal pressure with an external pressure, then there will be no variations in the pulse plethysmogram signal, right? So that is what is done, that using some feedback and fast feedback, an external signal is put, which looks like the which would actually look like the arterial blood pressure, because this is the force which is externally applied and that nullifies the effect of the arterial blood pressure. So the measurement that we get is a kind of pseudo measurement for the arterial blood pressure. So this is what is done in the Phenopress instrument. But what happens is that time to time we actually release the pressure to make sure that the system is in calibration, right? Because the person's blood pressure over the day, or due to various issues in the cycle of the control loops of the body, the blood pressure signal actually, you will see that you can say it is not a constant. Rather, it has some kind of an envelope. It follows some kind of an envelope, right? And there will be variations over the day and things like that. So one setting will not be able to control for the entire cycle over the day. So time to time there is a calibration done, right? So in that, at that time the feedback is released, right, or interrupted, and then we sort of we'll allow the control to again closed loop control to take over and then again synchronize itself, right? So whenever that happens, in between temporarily we'll allow a little bit of pressure variations, PPG signal, but even then after that, again we make it flat, right? So this is a very good measurement. Only thing is that it has these gaps in between. So we thought initially, in a naive way, we thought, okay, how can we fill up the gaps? Because this looks like some pattern is visible. So can we not figure out the pattern and then fill up the gap? So we tried to so this is the kind of signal that we get, right? So basically a few cycles of the ABP, they are missing, right? And as I said that there will be these patterns, right? So you can really see these patterns, right? So if you can figure out the patterns accurately, then right, you will be able to fill up those missing points, right? But as we tried to delve deeper, we found that it is not such a trivial problem. And so what we tried is that we just took the raw ABP signal, right, like this, and then we did a low pass filtering, right, and then we detected these cycles, right. So these first cycle, second cycle, third, like that. Each one we counted and detected those cycles, right. And then we identified segments where there continuous good signals are available, right? And then we identified the first and the last segment of good cycles of each segment, right? And then for each cycle, actually, we then figured out that if we have to interpolate, so the last cycle of the previous cycle and the first cycle of the next cycle, they have to be connected by some method. So we have all these segments, good segments. Now we have those pieces are available, and then we have to connect them through some means. So how do we do that? So because we have some information missing here, right? Right, and also we found out that even a person who's having a normal heart rate, these intervals are not exactly constant, right? So how can we find out the exact intervals? So we can do that from the ECG signal, right? So if we take the raw ECG signal, right, again, as I mentioned, these R peaks, etc. there, right? So gap between these will tell us what is the current interbeat interval or heart rate. Okay? So if we can get it from we detect the peaks in those the ECG signal, and from there we actually find out. So within this gap, right, how many such ECG signals will fit? How many complete ECG signals will fit? First, we find that out. So it will be 2, 3, 4, or maybe more sometimes, right? And the time periods of each signal, because we know the corresponding, these two must be synchronized, right? So from here we'll pick up those corresponding cycles, intervals in which we actually have to fit in the ABP signals. Then find out what is the number of cycles, number of samples, and then we can actually use some kind of a linear interpolation to start with, and I'll tell you that this is not really a very good method, but it's a good way to start, and then we can actually fill those up, right, by putting some kind of interpolation, right? So this is the algorithm we thought of, and which is still not perfect, because as I show you, so if you have this kind of a segment, right, theoretically you can fill up, right, but there is a disadvantage. In fact, I'm not sure whether you can figure out, but it is quite possible, especially if your length, the gap is large, it is quite possible that this could have come down and gone up, right, and then things done like this, right? In some way, it could have happened in different ways, because in this interval actually we have undersampling, right? So anything from signal processing, any place where you are doing undersampling, the variations could be happening anyway. In fact, we also found out that there are other effects, artifacts like breathing, right, respiration, or something which is called as the Mayer wave, right, which are some oscillations which just seem to be visible in arterial blood pressure signal, right? So these approximations, so far whatever we have done, this linear interpolation is not able to take care of it. So currently we are looking at incorporating these other features, right, which will help us in getting a better approximation, right? And basically it means is actually more deeper study about the physiology. So as I was mentioning, that signal processing, a lot of things look like signal processing from the outside, but once you go and try to dig deeper, you see that basically you need to go into the physiology of the persons and so on, right? So that will be the next topic. So how do you go and fill up these gaps in a more scientific fashion and by a deeper study, or we may call it deeper learning, deeper study, or modeling. So we would like to represent this as a kind of modeling exercise, right? So I'll just give you a very brief idea about how we model the cardiovascular system, and then how do we estimate the parameters in that system. So let me just, before I start, because on this topic, let me ask my hosts. So what is the time available for me? Because I seem to be going a bit slow compared to what I thought, and we're already around 45 minutes past. So what is the time limit?
Sir, we have 15 more minutes.
15 more minutes, right?
Yes. Yes, sir.
So, okay, then what will happen is that I'll probably stop after some time, and instead of trying to cover everything, I'll stop after talking about the cardiovascular system. And if people are interested, I can give a second talk, or maybe provide a recording of the rest or something like that. Okay, we'll come to that. Okay. Okay, so talking about cardiovascular system, I think all of you have studied, taken a look at the heart and the blood circulation in our body, right? So we have in fact two parts of the heart, right part and the left part. Right? So left part has left ventricle through which actually blood comes out and then goes through the entire body, right? And then finally they on return they actually so this is the arterial network, right? And this is the veins network, right? So the oxygenated blood, which is shown as red, goes to the arteries, and then they return through the veins, becoming blue. That is, they are deoxygenated, and then they enter the right part of the heart, and then they go to the lungs through the so-called pulmonary arteries, right? And they are still blue because pulmonary arteries are still carrying the deoxygenated blood. So this is still blue, right? And then they get cleaned up, right? They again get distributed into the various beds of the capillary beds of the lungs, and then they exchange through the gas exchange process. They exchange the carbon dioxide with oxygen, and then they become red. Right? So oxygenated part again comes to the right atrium, so left atrium, and then it goes to the left ventricle and then gets pumped to the body. Right. So this is the overall idea. Right. So we can have some different types of modeling of the cardiovascular system. Right. And so basically you can say the right part of the heart, the left part of the heart, and the pulmonary vascular bed, right? This part, right, where oxygen gas exchange is taking place. This is the right part of the heart, left part of the heart, right? And with a lot of simplifications. And overall, you know, systemic bed, right? The basically the body through which the blood is actually getting distributed. Blood is giving up oxygen and then getting carbon dioxide from different parts of the body. So this is how the various parts of the heart can be modeled, right? And just like any transportation process, you have this resistance to the flow, right? And capacities or capacitance to store the as reservoir of the flow, right? So, you know, a very simplified model could only have resistance and capacitance, but they are not adequate to represent all types of phenomena, right? So heart itself is like a time varying capacitor, and the beds are modeled with capacitors and resistors, and there are also valves, right? You probably know about the heart valves, right? There is from atrium to ventricle and so on. There are a lot of valves, mitral valves and things like that. So all of the valves are actually represented as diodes, maybe with a resistance in series, and some certain things are also ignored in the simplified model. So this could be a very simple model, and but even then, the estimating the parameters of all these resistance and capacitance is not a trivial matter. One of the reasons is that from the available measurements, you cannot actually estimate all the all the parameters. Okay, so our goal would be to estimate the parameters. So every a person or subject is coming, the patient is coming, and you need to determine his or her parameters, right? Then you have to make some measurements, and from there you have to estimate the parameters for that particular person, right? So you may need some estimation technique like least squares, especially nonlinear, because the models will be nonlinear. And then you will actually check whether those models that you are getting, whether they are actually making sense, right? And then, since as I said, all the parameters may not be sensitive enough, right? So you may need to do some kind of a sensitivity analysis, right, to find out which are the sensitive parameters. So I'll show you a case study where I'll actually explain these things. You can have various models of, you know, the earlier model was too complex. Here actually we just have a model of the left ventricle, right, and that of the systemic vascular bed. In fact, you can actually see that this could be modeled like a maybe with a third order model with just 3 capacitors, right? 3 capacitors will help us to get a third order model, right? Even these kind of models, they provide us a reasonably good estimate of the physiology, right? So what it can give us is actually it can, if we initialize it properly, it can give us the arterial blood pressure waveform, right? Or at least an approximation of it. And you have to initialize the parameters carefully with height, weight, and gender and other quantities, right, to get a good accuracy. And if we get an ECG signal, then we can actually use it for generating what is called as an elastance function, right? Basically, elastance is this quantity. Basically, the variation of this capacitance is captured by an elastance function. So you can actually model it as a function of the ECG signal. That gives us a reasonably good accuracy. or the model could be a little bit higher order, and unless it is a little bit higher order, certain phenomena cannot be captured. You can see here that here we have also put an inductor, right? So if you recall that once we mentioned about the ABP signal having a notch, right? And we observed that unless this inductor is there, we cannot actually get this notch in our model, right? So using that, let me show the results that we got, because that actually requires an oscillation, internal oscillations to happen. So we need LC oscillations for representing that. So some of the ECG signals, some of the ABP signal may not have a notch, while some of them may have a notch, right? And if you have an inductor, then it is possible to approximate with an inductor. Or the notch can be approximated with an inductor. Now, so we must. So what is the advantage of doing this modeling? Right? So we actually did a study on classification of coronary artery disease versus people having no disease. Can we really distinguish between them through the use of a model? Right? So we had set up a model. In fact, this model has been done by our earlier researcher Karan, Dr. Karan Jain, who actually provided this model. So what we did is that we fed the PPG signal, right, and the systolic blood pressure and diastolic blood pressure values. Remember, these are individual values and these are not the waveforms, right? Our target is to generate the waveform. So waveform will come here, right? Right. Of course, you see that here we have not used an inductor, so the notch will not be there. So more like it will be a flat kind of waveform, right? But even that gives us a lot of ideas about the behavior of the person, right? And to improve the classification, we also try to use something which is called as the metadata. So this metadata could be like, the gender of the person. I don't know why it's not coming inside. Oops. So this metadata, so there is some problem here. I don't know. Okay. Sometimes it is coming, sometimes it is not coming. Okay. Okay, so this metadata is actually age and other parameters, like, we check also whether the person is smoker or not and things like that. So using all of that, right, we actually enter them into a feature table, right? And then we actually get certain quantities from the model, right? And using that, we train the system with the 5-port cross validation and then do a testing, right? And then this is a closer view, right? So as I was mentioning that all the parameters, it's not possible to estimate, right? Only some of the parameters are more sensitive, right? And we try to estimate those more sensitive parameters, okay? And the remaining ones, we try to initialize them in an appropriate fashion, right? So this was a work published by Karan a few years back. And so we went further and tried to use it for detecting CAD, right? So some of the parameters which became relevant are these quantities, right? And some metadata, like, as I said, age, smoking, and whether the person has diabetes or chest pain and things like that, right? So using them, we actually could identify quite accurately, right, with a sensitivity of 97% and specificity of 93%, right? And so this was for the validation, and then test result, we actually got a little bit less sensitivity, but still, almost 90% result. And this is for a small data set that we actually got from our collaborators in TCS. where they did some experiment in Fortis Hospital. So this has been, I think, quite an enriching experience so far. So as I mentioned, that time may not be available for me to complete the way I had planned, because I still have ECG and PPG signal processing part is left. But since the almost 1 hour is over, let me pause here and let me take the questions. Then depending on the audience interest, then I can decide the rest. Or let me do one thing. Let me make certain concluding remarks. And also acknowledge the contributions of various people. So let me talk about the recent trends. Right. So probably I've already given a hint about the recent trends, that data fusion, like fusing signals or at different levels, right? We can fuse two signals, like ECG signal itself could be having multiple leads. Whether we can use more leads to get more information, this could be a very interesting question, and it's already being studied. Similarly, if we have cycles of ECG, multiple cycles of ECG, can we really create better signals from the multi-cycle by forming some kind of a templates? In fact, we have done some work on templates as well. And when there are multiple signals, right, we need synchronous data acquisition between them. Or we may actually fuse the signals at a feature level. That is, we extract features from the signals and then fuse the features and then use them for training. Or we may take a fusion, we will do a fusion at the decision level. That is, we get multiple different decisions from different classifiers, right? So from ECG signal we get one decision, and from PCG signal we may get another decision. We fuse them together so that we get a better decision, like a voting logic and things like that. So this is one thing. Another thing that is happening, which I have called as a recent fashion, because everybody is doing it, so it's deep learning. So a lot of effort is being put to apply deep learning techniques to get a better and more accurate classification. In fact, I had some results where I have shown you that I would have shown you that the classification using deep learning is marginally better compared to standard machine learning. But I think the last word is not yet out there. But there are different types of deep learning models, so they could be tried out. Right. So signal-based one-dimensional or image-based two-dimensional or even volumetric three-dimensional visualizations of ECG and other signals are being attempted to see whether we can actually get a better accuracy of our classification. And let me also conclude with the concluding remarks that now it's almost 400 years that William Harvey actually first showed that how blood flows in our body, right? Earlier, people used to think that the pure blood, the oxygenated blood, flows through the veins and returns to the arteries. But Harvey showed that it's the other way, right? And he did actually a lot of work and actually experimental work to prove that. So since then, a lot of modeling and other type of work have happened. But these days, there is a new opportunity with the new sensors and data collections, transmissions, IoT, and things like that. So, and then wearables and things like that. So, every individual person's health can be using this kind of data. So, if we have good algorithms, good signal processing, then we can actually detect it quite early, rather than waiting till the person is having a chest pain and then has to be operated upon. We can actually detect much earlier that the person's blood pressure or is going up and the ABP signal is not looking good and things like that. So we can actually figure out things much earlier. So that's what I meant. So early detection, if we can do early detection of any diseases, then we can not only prevent a lot of things like heart failure and strokes, but in fact we can actually alert the persons to maintain a certain cardiovascular health, and not without needing any other actions at the end. So with this, let me acknowledge some of our group members who contributed to the work. In fact, I could show only the work of Ashutosh and Karuna today, and of course Karan, as I mentioned, who is an alumnus of this group, and Sawan and Apurva, they are working on ECG, and Rajeshwari is working on PCG or phonocardiogram signals. Master students, they are all working in different signals. In fact, two of them are working on ECG signal. And one thing I would have mentioned in detail, if I got time, is that our team participated in a cardiological challenge from Physionet, and all being in a novice still, we came up 15th in a team where almost 200 people participated, and finally about 40 teams survived till the last stage. So our team not only survived, but it got the 15th position. And next year there is another upcoming challenge on cardiology. So our team is now very keen to participate. So a lot of people are interested to work on that. And I also mentioned about Rakesh's work, which is actually the interpolation of the Pinapress signal, right? So Rakesh is working on arterial blood pressure and PPG signal. And a lot of group members, new group members are there who have started working in internship and their BTech projects. And a couple of them have joined more recently. I think we have not got their photographs yet. So almost 6 people are working on ECG, EEG, right? And also Anshul is supposed to look at erectodermal activity signals. We'll see how it works. And some who have already graduated. So Karan and Vibhu, both of them are past PhD students. And Saiful and Gautami, they have been past master students who are now working outside. And then myself and Professor Nirmala Ghosh, we are trying to keep this group together. And so now we're ready for any questions. So I can even ask some of my students to answer in case there is any very specific details are needed regarding any part of the work presentation.